Greetings and welcome to the United States Transhumanist Party Virtual Enlightenment Salon. My name is Gennady Stolyarov II, and I am the chairman of the US Transhumanist Party. Here we hold conversations with some of the world's leading thinkers in longevity, science, technology, philosophy, and politics. Like the philosophers of the Age of Enlightenment, we aim to connect every field of human endeavor and arrive at new insights to achieve longer lives, greater rationality, and the progress of our civilization. Greetings, ladies and gentlemen, and welcome to our U.S. Transhumanist Party Virtual Enlightenment Salon. Today we have a special conversation in store for you about some emerging scientific research that could help to remove arterial plaque by clearing the non-degradable cholesterol that accumulates within cells in the arterial walls. Certainly, cardiovascular disease is still the leading cause of death, and if we want to overcome aging and death, this is a problem that needs to be fought with a lot of the available resources. So joining us today, we have our Director of Community and Citizen Science and 2024 U.S. Vice Presidential Candidate, Daniel Twedt. We also have Dr. Bill Andrews, who is our Technology Advisor and founder and CEO of Sierra Sciences. And we have our Legislative Director, Jason Geringer, joining us as well. And our special guest today is Dr. Matthew O'Connor, who also goes by Oki. He is the CEO of Scientific Affairs at Cyclarity Therapeutics. He was awarded his master's degree in neuroscience from Northwestern Medical in 1999 and his PhD in biochemistry from Baylor College of Medicine in 2005. His postdoctoral research includes work at UC Berkeley on muscle stem cells and aging. He is also the former Vice President of Research at the SENS Research Foundation, where he oversaw a broad swath of research projects spanning many aspects of rejuvenation biotechnology, from which he authored many papers and patents. So Cyclarity Therapeutics and its work will be the focus of Matthew's presentation today, and then we'll have time for a discussion, some questions, a deeper dive into that work. So Matthew, welcome. Thank you very much. And please feel free to begin your presentation.
Okay. Thanks so much, Gennady, and thanks everyone for coming to speak with me today. So thanks for the kind introduction. As Gennady said, I am co-founder of Cyclarity Therapeutics. And our technology spun out of the SENS Foundation, which is now merged as the Longevity Research Institute. But a few years ago, we moved up to the Buck Institute for research on aging up in Novato, California, which is where we are headquartered now. And one main point that I want to make big picture about our approach and about our technology is that we're remediating something that we believe is a fundamental basic part of the aging process, which is a part of oxidative stress, and oxidative stress to one of the fundamental building blocks of life, which is cholesterol. And so when we say that we're developing a cardiovascular disease drug, really we've developed a drug that targets a fundamental aging molecule, and we're targeting cardiovascular disease as a disease indication first. But I believe that our drug will have applications in many aspects of aging and aging biology and medicine. So to take a quick step back, the type of technology, the technology platform that we're using to develop next generation therapeutics at Cyclarity is these scavenging molecules. And so we developed these scavenging molecules to encapsulate small toxic biomolecules that accumulate with age in various cells and tissues, and then float away with them. And we settled on these type of molecule called cyclodextrins. Cyclodextrins are cyclic carbohydrates. They've been used in in many aspects of industry, chemistry, over the years, but most in the last couple of decades in medicine, they've been used mostly as delivery tools to deliver drugs as a part of drug formulations. But in 2015, the first cyclodextrin-based drug was approved, and it was acquired by Merck. It's now called Bridion, otherwise known as Sugemadex. And what it is it's an antidote or a reversal agent for a anesthesia agent, a paralytic. And so it grabs onto, after when you're going to get surgery, you get dosed with a paralytic so that you don't move around during surgery. And then the anesthesiologist gives you Bridion after they're done with the surgery, and the Bridion sweeps through, grabs up all of the paralytic, pulls it out of your bloodstream, and you pee the Bridion and the paralytic out, and you wake up right away, and you have no side effects from the paralytic, and you're better off. Bridion is a blockbuster drug. It sold $1.8 billion last year, which is more than all of anesthesia combined. And this is the same type of approach that we want to take with our drugs, is to make things that are specific for a certain target that you don't need any more of, right? You don't need any of that paralytic in your body after the surgery is done, so you just get rid of all of it, and the optimal amount of it to be in your body after the surgery is zero. So we developed this cyclodextrin technology, which is a dimerization technology where we stick two cyclodextrins together. And that's pretty much the core of our intellectual property, which is the ability to encapsulate whatever it is that we build to target with one of our cyclodextrin dimers. And we've been granted several patents in this area. So most of what I'll talk about today is targeting the oxidized form of cholesterol called 7-keto-cholesterol. I'll talk more about that in a minute. We're also working on targeting junk that accumulates in your eye with age and causes the major form of macular degeneration. We're also looking at nanoplastics, which is a good use case for our technology, as well as some other things like anesthesia reversal, which obviously there's many other anesthetics that we could build reversal agents for. So we started out pretty early on doing some pretty intense computational chemistry with cyclodextrins. And as opposed to other groups, which there's many groups doing computational chemistry simulations these days, not enough. I think it's a growing field and something that everyone needs to invest more in. But instead of going really general with computational chemistry or specializing in what most people specialize in, which is either protein-protein interactions or ligand receptor ligand binding interactions, we focused on only modeling cyclodextrins and their bonding with whatever target that we wanted to go after. And we do increasing levels of sophistication of simulations until we get to the level where we can actually predict an affinity constant for a given target before we even synthesize it. And now what we're doing is layering on AI and machine learning so that we can actually take the human out of the process of designing the drug and let the AI iterate until it comes up with the ideal binding partner for whatever kind of molecular junk that we're trying to get rid of. So we did a lot of that to come up with our lead drug candidate, and then we synthesized a number of prototypes and iterated back and forth between the chemistry and the bench until we came up with our first drug that I'll tell you a little bit more about in a minute. But first, let me take a step back and talk about the free radical oxidation process that causes the kind of oxidized cholesterol that we're worried about. So you've probably all heard of free radicals. Free radicals have been around aging medicine and biology for decades. And when you have an oxygen-free radical, they can react with a lot of different molecules in negative ways. They can react with your DNA, with your mitochondria. When they react with regular cholesterol, which is pictured here in yellow in the middle, then you add a oxygen molecule on, typically, so this is a non-enzymatic process, it's a random process, but it's not so random because more often than not, the chemistry prefers that it adds it in the 7 position, which means that you get 7-ketocholesterol, or 7-KC, and I'll sort of use oxidized cholesterol or 7-KC interchangeably in the rest of my presentation. And so oxidized cholesterol has been implicated in many aspects of aging, including aging in the eye, and liver aging, and brain aging. Most of what I'm going to talk about today revolves around how it causes atherosclerosis in your blood vessels, and how it does that. So a lot of the concept behind cholesterol oxidation and how it happens and where it happens and when it happens was worked out. A lot of it was worked out in the 90s in vitro and in mice. But it's only in the last decade that we've started to see some really convincing evidence that oxidized cholesterol is well associated with cardiovascular disease, with negative outcomes and with severity of disease. So there's a growing awareness of oxidized cholesterol's involvement in heart disease and atherosclerosis. So how does it do that? So when you get a lesion in your blood vessel wall, like a fatty streak that you start developing in your teenage years, a macrophage, part of your immune system, is supposed to come along and eat up that lesion. But when it runs into oxidized cholesterol, it can't digest it, and it can't recycle it. It can't transport it back out into the bloodstream. Now, macrophages are really good at recycling cholesterol. When they get regular cholesterol in the form of LDL, they take it apart and then repackage it into HDL, and if they don't need it, and then return it back into circulation, where it can go back to your liver for recycling back into the rest of the body, where you might need more cholesterol. But with the oxidized forms of cholesterol, it can't do that. It can't do that reverse cholesterol transport. And so it just eats more and more of it. And this is the oxidized cholesterol is highly toxic. And it causes the macrophage to start sequestering lipid and balloon up into something called a foam cell. And foam cells are these big blobby cells. They're kind of like senescent cells, but it's not caused by overdivision, like with fibroblasts or something like you might have heard of, but it's caused by toxicity from things like oxidized cholesterol. And foam cells build plaque. They are the driver of the mass of the plaque accumulation that you get, and it's an early and continuous stage in the plaque building process. What we wanted to do was ask ourselves, could we design a drug to specifically bind the common oxidized form of cholesterol? Could we remove it from cells and tissues? And could removing that reverse the foam cell phenotype and potentially rescue atherosclerosis? So we built this drug, which we're calling UDP-003, which we trained first computationally and then synthetically to prefer binding oxidized cholesterol and to ignore regular cholesterol, which you need to survive. And so we built this first drug, and it has a very high affinity for the oxidized form of cholesterol, and it leaves regular cholesterol alone. And we've done a great deal of work to characterize this drug and get it ready for to be a drug. So first, up here at the top, you have macrophages, just normal healthy macrophages grown, and then you give them a little bit of oxidized cholesterol, and they balloon up into nasty-looking foam cells. And then when we give our drug, they turn back into healthy macrophages without those blobby lipids in in their cytosols. And here's quantification of that here.
So if you go back for a moment, I just want to see the quantities here. So it's 10 micromolar, 7 keto cholesterol. Yes.
Yeah, in this particular case, we've done it many different permutations of this, many different ways, and you can see how that's working here. And so that's just how the macrophages look, how they look better. They look younger, right? They were nasty and old looking, but do they go back to doing their job? And the answer is yes. If you poison them with the oxidized cholesterol, their ability to eat, their ability to do phagocytosis drops. And then when we give them our drug, the phagocytic capabilities come back. Multiple different ways with both human and mouse cells that we did this with. Perhaps more importantly, I mentioned earlier reverse cholesterol transport, and that's a big part of the job of the macrophages in your blood vessels is to recycle, is to get rid of the excess cholesterol. But when you give them oxidized cholesterol, like over here on the left, the reverse cholesterol transport activity goes down to almost zero. And then when you give our drug, just very briefly for a short amount of time, it goes back up quite significantly, which we think means that our drug is restoring the ability of the macrophages to do their job, not just in eating, but also in metabolizing cholesterol, transporting cholesterol. And that's a pretty key aspect of atherosclerosis. So then we went through and did a good amount of characterization of the phenomenon and looked at gene expression. And I'll just show a few examples from a recent publication on this. One is the CD47, which is being targeted by a competitor of ours with a therapeutic antibody, which is kind of like in a tumor where the tumor evades the immune system. The foam cells do the same thing. So they turn up this don't eat me signal called CD47. And what we've shown is that our treatment with our drug brings that back down a bit so that the cells should be able to be cleared. The foam cells should be able to be cleared by other macrophages. Another various aspects of lipid transport get downregulated and then go back to normal when we treat with our drug, and then various other aspects of inflammation, of scavenging genes that are either upregulated or downregulated in response to the insult, which is the oxidized cholesterol, and then get reverted variously by treatment with our drug. Out of the weeds a little bit, what happens to animals when you give them our drug? Well, it's still early days with really fully investigating this, but this is the most exciting data that I think generally that rodent models of cardiovascular disease, of atherosclerosis, are pretty artificial and unrealistic. And so I don't really like them, and I don't like working in them. But in normal, healthy rats, if you give them our drug is designed to be cleared very quickly and to be cleared through the urine. And that's exactly what we see. I'm not showing the data here, but actually, when we dose with our drug, we see the target of our drug, which is the oxidized cholesterol, coming out in the urine, and we see it coming out in the same time scale as we see our drug being excreted in the urine. And so this suggests that our drug is doing its job, and it's doing it very fast. And so the animals are peeing out their oxidized cholesterol within a couple of hours of administration of the drug. And so this story that I just told you was just recently published in the journal Atherosclerosis. And I should add in here that we just, a professor in the field, without talking to us about it, wrote a nice commentary about our paper, and that was just published this past week. We wanted to turn this into a clinical stage drug. So we worked on the chemistry and the manufacturing. We scaled the process to kilogram scale manufacturing. We made large scale batches. We put it into single-use sterile vials. We had many meetings with regulators in many countries, in the UK, in the US, and in Australia. And then early this year, we launched our phase 1 clinical trial in Australia. And I'll tell you more about that in a minute. But first, here's the leadership of our team, me and Mike Cope, the co-founders of the company, Noah Rosenberg, the chief medical officer, and the clinical advisory board that he recruited to help us turn our drug into something that could go into people. So PK Shah was the main guy who dissected in the 1990s the cholesterol oxidation process, and Steve Nichols is arguably the world's expert on how plaque is built. He's got about, I think, 800 publications on the subject, and he is leading our clinical trial now in Australia. So we're doing what's called a traditional SAD-MAD trial, which is single ascending dose, multiple ascending dose trial. So we've just completed the single ascending dose portion of our trial, and so healthy volunteers have received the highest dose of our drug that we intend ever to give to humans. It's preliminarily seems to be safe. And now we've started in the first stage of the multiple ascending dose. And one innovation that we're doing is our phase 1 trial is being run in Australia, which isn't particularly innovative. Probably a third of all phase 1 trials are being run in Australia these days because it's a great place to run early stage clinical trials. But our trial is actually, instead of being run by a private company, it's being run by an academic research organization, the Victorian Heart Institute, which is headed up by Steve Nichols. He has the country's foremost heart hospital, and we're super lucky. This is the type of guy that the big drug companies call to get their drugs over the final stages, like the Lp(a) drugs in phase 3. He's worked on those, bempedoic acid. He helped get those approved. So to have somebody like him taking an interest in our drug at such an early stage is really, we're really lucky to have him. So a little bit more detail about the design of our trial. We started out at an extraordinarily low dose of our drug, only 1/25th of what we were eventually intending to dose, and scaled it up until we got to where we are now, and we've just gotten past, which is the highest dose, which is 25 milligram per kilogram of the recipient. And like I said, we've started the multiple dose. So, of course, the main outcome that we're looking for is safety, vital signs, reactions, hearing tests, looking at the pharmacokinetics, how fast the drug is cleared from circulation. And then what I'm most excited about, and the data that I'm really looking forward to seeing in the coming weeks, is the pharmacodynamics of the target engagement, to see if our drug is actually grabbing the oxidized cholesterol and pulling it out of the human patients, out of their blood and into their urine. So we already have permission. Another nice thing about doing trials in Australia is that they're quite flexible with trial design, and they've already given us permission. Once, if our drug is proven to be safe in healthy young volunteers, we can go straight into a what's called like a phase 1B type trial in a small number of human patients who have coronary artery disease. They have plaque in their coronary artery. And we can dose them with our drug. We can look for that target engagement, see if they're peeing out the 7-KC. And then we can look at blood inflammation markers, which we'd expect to go down based on some of the data that I showed you before. And we can do non-invasive imaging of their plaque using CTA angiograms, look at their plaque before and after treatment and see if it improves, see if it gets reduced, see if it looks less inflamed, that kind of thing. Next is our phase 2 plan, which is a scaled up version of exactly what I just described for late stage phase 1, which would be in about 150 patients who already have acute coronary artery disease. And we'll look again at the oxidized cholesterol in their blood and their urine. We'll look at their blood work, see if it improves, and do the imaging. And then, of course, we'll monitor them for heart attacks and strokes. And hopefully that gets better over the short duration of the phase 2 trial. So I want to take a little bit of a step back before I wrap up and talk, just remind you what I said before, which is that oxidized cholesterol plays a role in brain aging because where do you have the most cholesterol, the highest density of cholesterol in your body? It's in your brain. So we're looking at using our drug for various aspects of aging and dementia. So we're looking at Alzheimer's disease, we're looking at stroke recovery. And like I said before, we're interested in macular degeneration, and we've recently become interested in looking at liver failure. So we've raised a good amount of money so far to run our phase 1. We're closing on a round of that now, and then we're going to start raising money for our phase 2. So if anybody listening is interested in investing in Cyclarity Therapeutics, please reach me. And we're expecting to make a deal with a large pharma company post phase 2, where we would license out our drug UDP-003 and basically hand it off to a large pharmaceutical partner to shepherd it through phase 3 clinical trials and approval and marketing and all of that. And at that point, we expect to have a big return for our investors in 3 or 4 years. So this is the team of amazing people that make this happen at Cyclarity here in California. But we've actually got a team of over 100 collaborators and consultants and CROs all around the world, and many too many to thank here. So I'll stop, show my disclaimers, and I can stop here and take any questions that you all have.
Yes, thank you very much, Matthew, for that very informative presentation. And we do have some questions from our panel thus far. So Daniel Twedt wonders, is injection of your drug UDP-003 the only administration route so far, or are there other possibilities as well?
There's various aspects of injection that we're looking at. So all the data that we have so far and all the work that we've done so far is intravenous, but we've tested it in animals subcutaneously, and it works very well. And so we're envisioning that it will eventually be released as either a subcutaneous or intramuscular injection. It could be like a patch or a pump. It's unlikely to be a pill.
Yes, thank you for that answer. And Bill has 2 questions. The first question is, why CCTA instead of CIMT? And Bill, I'll let you elaborate on that question for the laypersons in the audience. Just a moment, Bill, you need to unmute.
Why a CT angiogram instead of a CIMT, since you're looking at the plaque before it's ruptured?
So the CIMT is generally done by ultrasound, and CTA is done by X-ray. Just for the audience, I know you know this, Bill. And you get a better resolution with CTA, and you can look more deeply into more hidden blood vessel walls, as opposed to being somewhat restricted to more surface level blood vessels with ultrasound. So the most quantitative, and this isn't all coming from me. It's mostly from my cardiologist, who insists that CTA is the gold standard right now in terms of doing really quantitative measurements, down to the single % difference measurements in plaque thickness, and also being able to see the fat attenuation index, which is how dense the plaque is, which tells you a lot about the characteristics. So even if the plaque volume isn't changing, you can tell if the plaque is getting ready to rupture, or if it's getting less likely to rupture. And you can also tell things about the inflammation using AI layered on top of the CTA imaging these days. So there's a lot of data you can get out of the CTA. The bad thing is that you're exposed to X-rays and it's not as fast and cheap. I wish we were doing both, and I'd like to move into doing both at some point in the not too distant future.
Okay, I was misinformed, I guess, about CIMT. I know you can only do it on the carotid arteries, but I was thinking that.
And people are less interested. I get into arguments with vascular surgeons and such about this. I think the carotid is a great place just as a marker to look to see if a drug like ours is working. But they say, "Oh, why do you want to look at the carotid? It's so easy to do surgery and fix the carotid artery. So there's no market for treating carotid stenosis. Just go after a major artery like the coronary artery." And I agree with you that it's a really nice, easy place to look for plaque and see it really easily and non-invasively. But I get kind of shouted down by the cardiologists when I try to make that argument.
Okay. I was actually super impressed, so I take your word for it. PCSK9 inhibitors with statins have been shown to turn soft plaque into hard plaque. Is hard plaque going to be still filled with oxidized cholesterol? I'm not sure about that. And will your cyclodextrins get rid of oxidated cholesterol in hard plaque, if it is there?
Yeah, I have an answer for you. I don't have a perfectly scientifically satisfying answer for you. I was really worried about that when we first developed our drug, about whether or not it was going to be able to penetrate plaque at all. And so we got samples of hardened plaque tissue from patients that it was taken out of surgically. And we soaked our drug in hardened plaque tissue, and it pulled tons of oxidized cholesterol out of the hardened plaque tissue. I don't know the mechanism by which it penetrates so well. There's not a good explanation for why our drug penetrates hardened plaque tissue well, but in our hands, very preliminarily, it looks like it does. And so I am optimistic that it will in patients. But it's something we need to look into more. And I wish there were better animal models to look at that kind of question.
I was super. So the other question I had was just combined with the first one. I was super impressed. I was asking these questions because I'd been led to believe from some clinical studies that I'm trying to get going that these were the top things to be doing. But I'm actually thinking what you're doing here might be better than PCSK9 inhibitors with statins for reducing plaque.
Yeah, I mean, the problem is that those are what's available today, right? So what's on the market now are LDL lowering drugs, statins and PCSK9 inhibitors. And the PCSK9 inhibitors might be working slightly better than statins, but they're both doing basically the same thing, lowering your LDL cholesterol, which has, this bad reputation as being the bad cholesterol. But you actually need LDL cholesterol. It's how you distribute cholesterol through your body. Most of the cells in your body wouldn't have any cholesterol if it wasn't for LDL cholesterol. So you can't just get rid of it. That's why I like the oxidized cholesterol as a target, because the optimal amount of it in your body is 0. If I could get rid of, it's like lead, right? What's the optimal amount of lead in your body? It's 0. So if you could just get rid of all your lead and all of your oxidized cholesterol, you'd just be better off. And so that's why I like it as a drug target. And I think it's it's a progression. It's the next generation. We're going to start seeing the Lp(a) drugs coming out, I think, in the next year or two, but those are only going to work on, it's basically a different kind of LDL cholesterol. And I think to really move the needle towards reversing disease rather than just slowing it down, which is basically what the lipid-lowering drugs do, we're going to need something more aggressive that can reverse disease.
So did you just say you're targeting, you have something that's going to be targeting Lp(a)?
No. I was just saying the next big heart disease drugs that are going to be released, I'm predicting, because there's several big drug companies that have them in late stage phase 3 clinical trials, and they seem to be working reasonably well, are going to be Lp(a) lowering drugs. And we will be the next generation of drugs after those, I believe.
Yes, very interesting. So that gives rise to a hypothetical question from me. So if cholesterol exists in the bloodstream, but it doesn't get oxidized, if you take care of all of the oxidized cholesterol, does the non-oxidized cholesterol pose any risk of atherosclerosis, or can the macrophages that you mentioned digest any other cholesterol to render it harmless?
Yeah, I mean, that's a big part of the job of the macrophage is to collect the cholesterol and recycle it, send it back to your liver. So that's what should happen. The issue is that you always have oxygen free radicals around, so it's like rusting. It literally is rusting, right? It's the rusting process of oxygen reacting, in this case with cholesterol instead of like metal on your car or something like that, right? But it's a similar process, and so it's going to keep happening. And so if you have elevated cholesterol, and you have the same amount of oxidation, if you increase either oxidation or cholesterol, you're going to end up with more oxidized cholesterol. So people with elevated cholesterol tend to have more oxidized cholesterol in their system. It's not a one-to-one, because you can have more oxidation in your system for various reasons. And we don't exactly understand why there are people walking around with like normal levels of cholesterol, but really high levels of oxidized cholesterol.
And is it possible to have the opposite scenario where someone has high levels of cholesterol overall, but low levels of oxidized cholesterol for whatever reason? Or do we not observe that unless that person has been treated with something like UDP-003?
You know, there are people who do seem to be somewhat resistant to walking around with high cholesterol. I don't think it's well studied enough to say that they aren't accumulating oxidized cholesterol in various places in their bodies, whether or not they develop atherosclerosis at a young age, like most of them do. I think they're probably building up oxidized cholesterol someplace where we're not looking, like in the brain, which is really hard to biopsy. You can only look at it pretty much after somebody dies, and other tissues as well.
Yes, thank you for that answer. And Bill has a question. Do antioxidants reduce the levels of oxidized cholesterol, or can some antioxidants do that?
You know, it's something that some groups have looked into a little bit. I mean, mostly antioxidants have been kind of a disappointment in general in medicine and anti-aging medicine, right? They haven't proven to be very effective. Now, the counter argument to that is that we haven't built good enough antioxidants yet. We haven't targeted them to the right places or something like that. So I think there's potential room for that. But you can't reduce your oxygen free radicals to zero. You can't completely prevent all oxygen free radicals from forming, no matter how many antioxidants you take. So I mean, that's why I follow this kind of damage repair philosophy approach. Generally in my approach to studying and trying to remediate the aging process and therapeutically is that I think it's the best place to intervene. But there is potential to see. I haven't seen yet a convincing study showing that there's good antioxidant evidence showing reduction in oxidized cholesterol levels in a person, but I don't think it's been studied enough. And I think better studies with better antioxidants could work to some extent with it. And perhaps we've started a small project looking at some kind of combination therapy in that realm in a model system that we're looking at, that maybe it could be helpful, especially since when you have high inflammation in some place like a plaque, you also get high levels of oxidation. And so if you combine antioxidation with getting rid of oxidized cholesterol, maybe we'll get a synergistic effect.
Very interesting. Thank you very much. And we have a number of questions from Daniel Twedt as well. Let's start with the questions that focus on the name of the drug. And Daniel is curious about the origins of the name UDP-003. Is that a chemical name, shorthand of some sort?
It's so UDP stands for Underdog Pharmaceuticals, which was the original name of our company. And then we changed our company name to Cyclarity Therapeutics a few years ago. So we did the kind of boring but efficient thing, which was numbering our compounds from starting from one as we invented them. And yeah, it'll get a more exciting name at some point. I didn't want to give it a sexy name too early, because what I see happening is that you get your favorite name and you name it, and then you get a partnership or you get a new big investor and they don't like your name and they make you change the name. And then you see drugs go through like 3, 4, 5 different names before they actually get, widespread on the market. And I figured I would just wait a little while before we, get married to a name. But if people have ideas for names for us, send them to me, and we're starting to make a list.
Yes. So for those viewers watching this Virtual Enlightenment Salon, please put any ideas you have for a name for UDP-003 that might eventually be used in our YouTube chat, and we will relay them to Oki. And in the meantime, Daniel has another question. He notes this scavenging therapy molecule sounds like it has benefits for both new as well as long-term users. And he wants to know if that's correct, given that this process of oxidation happens continuously. So would you expect that this is a drug that could also be administered over the long term for someone to prevent the formation of atherosclerotic plaques?
Yeah, I think it can be a preventative as well as a reversal agent. And I hope we see a day when, based on a biomarker test like oxidized cholesterol in your blood or in a tissue sample, you can take our drug, and that you don't have to wait until one of your blood vessels is like more than 50% occluded when you start having trouble, climbing stairs or breathing or getting chest pain or something like that, or having a heart attack before you ever get diagnosed with having atherosclerosis. So yes, I think it can be a preventative as well as a reversal. I don't think that you will need to take it continuously, like every day or something like that. I think you can take it intermittently. And we hope that you will just have to take approximately 6 doses in a row over a couple of weeks. Our current protocol is over the course of 16 days. Every third day, you get a dose, and that should get rid of the vast majority of the oxidized cholesterol in your system. And that's taken decades for it to build up. So hopefully you don't have to come back for another 5 or 10 years for another course of treatment. So maybe people are going to start getting this when they're symptomatic in their 60s, say, but later on, maybe we'll be taking it in our 30s or 40s every 5 or 10 years to make sure you don't get sick from that stuff.
Yes, indeed. And would you pair that with some sort of diagnostic test, say a blood test to measure the oxidized cholesterol, the 7-keto cholesterol, rather than just the total cholesterol or the LDL?
Eventually, we're not developing there's not a commercially available 7kc. There's two kinds of blood tests you can do for oxidized cholesterol. One is for oxidized LDL, which there is a test for, and you can go and get. Our drug should not directly remove ox-LDL. Our drug isn't meant to attack LDL or HDL. It's meant to attack cells and tissues. So what ox-LDL does is it delivers 7kc to the blood vessel walls and to macrophages. And so then our drug can go into the macrophages and pull it out after it's been delivered. So you can get a test for ox-LDL, but our drug shouldn't be lowering ox-LDL. We can test in our clinical trial, we are testing people for blood and urine oxidized cholesterol, 7-ketocholesterol, but we're not developing a diagnostic that we're intending to release and sell to the public. I think that the world will be better off if and when there is such an approved diagnostic. We're doing a little bit of work trying to invent a faster, cheaper method of measuring 7-ketocholesterol so that us or somebody else, a diagnostics company, could take that and turn it into a diagnostic test that patients could take. To your question about what it will be paired with when it's first released, it will probably be paired with angiogram. So you'll get a scan of your say coronary artery, and it'll be blocked, and you'll take our drug, and you'll come back in 6 months or a year and get another scan, and hopefully it will have improved. And maybe it'll have improved, but it won't have improved as much as your cardiologist wants, and so they'll give you another course of treatment, and they could do that, once every year or so until they're satisfied that you're better. But eventually I hope that it gets reduced to an easier biomarker like a blood test.
Yes, thank you. And that leads into the question from Daniel Twedt. How affordable would this therapy be envisioned to be?
Well, we expect it to be covered by insurance. We're planning to release it, widely in all the markets around the world. It's not going to be, $1 or $10 a treatment. It's probably going to be at least a few $1,000s for a treatment, and we don't know the market economics yet. It's probably going to be thousands of dollars for a treatment. But I think it's more expensive than like a statin, but it's going to be cheaper than a gene therapy and competitive with like therapeutic antibodies.
Yes, and as you said, hopefully the few $1,000s per treatment will be administered every fairly large number of years, like 5 or 10 years, rather than, say, every year. So thank you for that answer. And Bill also wonders, with regard to the comment about the angiogram, will UDP-003 reduce clots also, or at least have a tendency to do that?
Let me say, I was thinking differently when I asked that question. I was forgetting that regular angiograms can detect large plaque bulges too. So, but yeah, is there any chance that this would help with ruptured plaques when a clot gets formed?
Our drug acts quickly in terms of removing oxidized cholesterol, but it acts slowly in terms of allowing the macrophage then to go back to doing its job and cleaning up the plaques. So our drug isn't designed to work like Drano and flush out your arteries in minutes. It's meant to flush out all your oxidized cholesterol and then for it to heal over the course of weeks and months. So you wouldn't want to rely on our drug if you were like in an ambulance needing like a clot buster, because the blood flow to your brain has been blocked. That said, there's clotting there's clotting that happens inside of plaque, and ruptured plaque can get stuck places where it's not very easy to remove surgically. And, sometimes there's decisions about whether to leave it alone or monitor it or go in and remove it. And so in some of those edge cases where where a wait and see approach might work better, then maybe our drug would help. But I'm kind of rambling because I'm thinking about this on the fly because I haven't thought about that question before, Bill.
There's another question I had, and that's because I was doing some Google searches while you were answering some other questions. But on cyclodextrins, I saw that there's 2-hydroxypropyl beta cyclodextrin that's already available for getting rid of oxidized cholesterol. Is yours comparable to that? And it didn't look like that's used as a dimer like yours is.
Yeah. So the 2-hydroxypropyl beta cyclodextrin, or just hydroxypropyl beta cyclodextrin, or HPBCD, it's been around for decades. It's used in foods. It's used I think it's in Febreze. It's used to deliver drugs. It's very safe. You can inject it at very high doses. You can eat it. There's food grade hydroxypropyl beta cyclodextrin. So should you get injected with it in order to get treated, say for aging or atherosclerosis or something like that, or other diseases. There are actually 2 companies that are trying to use that substance to treat Niemann-Pick type C disease, which is a rare lysosomal storage disorder, which is a cholesterol transporter disorder. Now, hydroxypropyl beta cyclodextrin doesn't bind cholesterol very well at all. It's a very, very weak, if at all, binder of cholesterol. But they figured this stuff is so safe and it's approved for like anything that you want to use it for. So we're going to take these kids and dose them with like enormous quantities of this stuff and see if it helps. Because it did help in animal models. So if you give an animal with this Niemann-Pick disease this hydroxypropyl beta cyclodextrin, when they're very young, it helps. It does help a lot. I suspect it is helping the patients. It's been a little difficult for these companies to prove that it's helping because these patients are so sick and have so much brain damage that it's hard to tell if they're getting better. I think they're living longer with a very poor standard of living. I think the drug is very weak, but probably effective at extraordinarily high doses. And the kind of doses that you need to make it effective, since it's such an inactive compound, is like having one of these kids trying to lie still on a table, getting a IV for like 6 hours or something like that. So I think our drug would work better for Niemann-Pick disease. We're not working on that, but I'd love to be working on that someday. And I think it would be the same kind of thing. If HPBCD could help you with atherosclerosis, I think you would need to be getting an IV bag full of it frequently in order for it to have an effect. It's just not a drug. It's something that is a very weak binder of a lot of things. So I don't know what else it's binding when it's going in there, but it's pretty safe stuff. So it probably doesn't hurt, but it's been shown to cause hearing loss at very high doses in rodents. So that's something that some people are concerned about. You just need to stay clear of those hearing loss toxic doses.
Interesting. Thank you very much. And we've got a few questions from Daniel. He wonders, could this be used to externally process a patient's blood in a kind of dialysis modality, or would that not make sense? I think, Daniel, what you're considering is perhaps taking a patient's blood and applying the drug to it outside of the patient's body and then doing some sort of like therapeutic plasma exchange where you pump in the clean blood and replace the existing blood with it. Or would that not really take care of the problem, which would be that cholesterol accumulating in the arteries?
Yeah, it's a good question, and it's something we thought about for a while. So one problem with the approach is the last thing that you just said, Gennady, which is that you could pull all of the oxidized, using a method like that, you could pull all the oxidized cholesterol out of the bloodstream, but you wouldn't be delivering your drug to the system and letting our drug go into your blood vessel walls and any other tissues where it might be able to pull oxidized cholesterol out of. Now, that doesn't mean we haven't thought about it and that it might not be a good therapeutic approach. What we haven't been able to think of a way to do is, so, you take the drug, or you take the blood out of the body, and then you put our drug in there. Presumably, you want to pull the drug out before putting it back into the body, or else, why did you go through this process rather than just delivering it into the body? So we haven't been able to think of a way to clear our drug out of the bloodstream as a part of that process yet. If there's people listening who are interested in how that might work, or think they might understand something about the how to make that work in a kind of a sort of a dialysis sort of approach, we're interested in that as approach, but we haven't started playing with it yet. But it's a cool idea that we want to work on sometime.
Yes, thank you very much for that answer. And Daniel's other question is, could targeted cyclodextrins be created using a suitably genetically engineered cell, such as perhaps an E. coli bacterium that could be harnessed for that purpose? Could you reconfigure one of those to produce the cyclodextrins?
So the core building blocks are built by microbes, but I've never heard of a microbe building a modified cyclodextrin, like the example that Bill was asking me about earlier is a hydroxypropylated cyclodextrin. So you take a hydroxypropyl chemical group and stick it onto a cyclodextrin, which is part of the power of cyclodextrins, is how you could, there's potentially hundreds or theoretically infinite different types of chemical groups that you could stick onto a cyclodextrin. And then a cyclodextrin, there's 3 different sizes of cyclodextrins. The cyclodextrins that we're using are the medium size, and we're making dimers of them. Our dimers have 40 sites on them where you could potentially put any of these chemical groups that you want. So they're kind of infinitely engineerable. Now, I've never heard of a microbe figuring out how to do that chemistry on its own, but they're very good at producing the core building blocks, and that's how the building blocks are made that we buy and then use to do our synthetic chemistry on top of that. But I've never heard of somebody figuring out how to culture, how to grow substituted, we call them substituted cyclodextrins, in a vat.
Mm-hmm. Yes, thank you for that answer. And Daniel wonders, a dimer is a chemical pairing of identical molecules. Yes, that's the definition, essentially a molecule or molecular complex consisting of 2 identical molecules or subunits that are linked together.
But you don't necessarily, so you could have the dimer, and you could imagine that on one side of the dimer, you have one kind of chemistry, and on the other dimer, you have another kind of chemistry. And we can do that kind of chemistry when we engineer our dimers. And that is part of our intellectual property, is making the 2 different halves different, so that if you have an asymmetrical target, you could engineer one side to grab the one side of your target in a certain way and the other side, so you could work on the charge. So if you're looking at a charged target, you could put the opposite charge on one side of the cyclodextrin and not on the other side. If your target is has a skinny and a fat side like that, you could make your cyclodextrin more narrow on one side and fatter on the other. Those are all things that we play with a lot when we're in the early stages of engineering our cyclodextrins at the computational and then synthetic level.
Very interesting. So the dimers are more versatile than they may appear at first glance, at least with your approach. Now, one question that I'm curious about is in regard to your future plans in the event that the phase 2 trials are successful. You stated you would make a deal with a large pharma company to essentially manufacture and sell this drug. Would you plan for a deal where Cyclarity would maintain the intellectual property and then the pharma company would pay you royalties over time to use it? Or would you just sell the intellectual property in one lump sum and let the pharma company do what it pleases thereafter?
There's a lot of potential ways that could play out. And, I may or may not be in a position to get my perfect wish. But my perfect wish is that we would do a partnership with a large pharma company, either a joint venture where we share responsibility for the further development, for the carrying out of the phase 3, so that we could have influence over how the phase 3 is developed, what kind of patients it gets used in, how it's being tested, so that they don't mess it up, that kind of thing. And then, going into the marketing, likely the big pharma company would do all the marketing, and then we would get royalties from that. So we would get upfronts and royalties, likely in either the scenario where it was a joint venture or where we just sold the drug to a partner and they just took it over from there. And then, another possibility is that our whole company would get acquired by a large drug company. That's not really my favorite idea, because what I really want to do is take the proceeds from that equity event when they buy the drug from us, so they do a deal and we get upfronts, and then we pour that money back into the company, into our platform, so that we can develop our next 10 drugs with those funds, which might not happen if we get acquired.
Yes, indeed. I think it would be a preferable outcome if you continue to have input and involvement and an income stream from how that drug gets developed and used in particular, because then you could ensure that it actually gets rolled out to patients. Sometimes pharmaceutical companies will acquire intellectual property and sit on it, and that's not necessarily the best outcome for patients who could benefit from the drug in clinical settings. Bill wonders, would UDP-003 qualify for fast-track clinical studies?
I think that to be fast-track, it would have to be rare disease. I'm envisioning it more angling towards accelerated approval, which is what FDA uses for chronic disease indications, where you may be able to get like a tentative approval and then need to keep doing studies to prove that you're having the outcome effects, like effects on heart attacks or strokes. That's really my dream for this, is that we would get approved on the basis of plaque regression rather than doing what is traditionally done, which is do like a long 5-year study with thousands of patients waiting for people to have heart attacks and strokes. And then you count bodies and you see if people on the active group die less or have fewer heart attacks than the people on the placebo group, which I think we're going to look back on those kinds of studies someday and think they're barbaric. I hope that we can get accelerated approval on the basis of their plaque regression and then show the reduction in heart attacks and strokes in the following years after initial approval.
Yes, indeed. That would definitely be more humane and better for the patients. So I know we're past the hour. Do you have time for a couple more questions, just the ones that have been posed?
Let's say 2 more questions and then I got to run.
All right. So Daniel wonders, is the Victorian Heart Institute funding most or all of the trials now taking place?
No, Cyclarity Therapeutics is funding 100% of the trials taking place. Almost all of the money has come from private investment into Cyclarity Therapeutics.
All right. And Daniel's comment, after which I will pose his last question, is this is almost like a precursor to what cellular nanomachines might one day accomplish. And I hope we all live to see the day when that happens. And his final question is, he sees that it's interesting that the blockbuster drug Bridion outsells the anesthesia drugs, or did he hear that incorrectly? Does it actually outsell the common anesthesia drugs?
Yeah, it outsells all of anesthesia put together. Now, that said, that's a little bit misleading because Bridion is still on patent, and so Merck can charge a lot of money for it, whereas most traditionally used anesthesia agents are generic and pretty cheap. But it's still pretty impressive to me that this one reversal agent, it's so incredibly valuable and profitable because anesthesiologists love it. It's incredibly safe and effective, And even if they accidentally or on purpose, anesthesiologists are getting lazy in dosing their patients with an excess of Bridion. And there was a study done recently trying to bring them to task and looking for the consequences of giving your patients too much Bridion. And they couldn't find any consequences from giving patients 2 or 3 times as much Bridion as they needed to clear out the paralytics. So the anesthesiologists are just shrugging their shoulders and not doing the math to figure out exactly how much they should give their patients and just taking a bunch off of the shelf and giving them the whole vial rather than calculating how much they should be giving their patient, because it's a next generation drug. It only grabs onto something bad that you want to get rid of, and then you pee it out, and you pee out whatever extra that you get. So it's really a perfect drug, and that's how we want our drug to work as well.
Yes, that sounds like a great drug, essentially no adverse side effects. So thank you, Matthew, for all of your answers and for your time today. That was a fascinating and enlightening presentation. Do you have any final words for our audience? Any comments that you wish you could have made, or anything else that you want to say before we close?
I just want to say thanks so much for having me here to present to the Transhumanist Party. And people have more ideas, whether they're about the name or about the science, or if you're in science and you want to collaborate with Cyclarity, if you're in a research lab and you have a model system that you think our drug could work in, we'd love to collaborate with you and get to the finish line together. It takes a huge number of people to get a drug approved, and there's so many wonderful supporters out there, and I invite everyone to get in touch with me on LinkedIn, follow Cyclarity on social media, and we'll try to keep you all updated on how things are going.
All right, thank you very much, Oki, for your time today. This was quite an enlightening presentation. Please, to all of our viewers, share your ideas with us in the chat, or reach out to Oki and to Cyclarity on social media, and I hope that we can all live long and prosper.
Thank you.
Indeed. Thanks.
Cody and Gennady. We want to get rid of this guy. As you know, our purpose here at RAADfest, Revolution Against Aging and Death, is to kill death, right? So, Joe, how are we going to kill death?
I mean, together. That's the whole thing. Together, in a coordinated, collaborative fashion, where we feel like we feel our life more than we feel that.
Okay, and Cody, how are we going to kill death? How is Immortalis going to kill death?
Well, everything Joe just said, but also we need to free the geniuses of society who are working on the science end of it, and we do that together.
Good. So, Gennady, tell us your view.
We need to get the public to recognize that killing death is both feasible and desirable. They need to see a plausible path for getting from here to there. And that's actually one way in which games like LEV the Game can help to get them to see it's not going to be a single moment in time. It's not going to be a single magic pill or panacea. It's going to be a process. But it's a process that each person, even if you're 65 or 75 or 80 or 90, has a chance in undergoing successfully.
Okay, I will play devil's advocate, and I will ask Death. Death, what do you think about that?
I was quite inspired by Joe's talk, and it's been quite lonely as the only ageless character. Maybe you should join me.
Wow, that's good. That's good. But how about if we killed you?
That would not be pleasant at all. Although, I am so busy of late, I could use the rest.
Good. At least we could freeze you, okay? We can keep you frozen forever.
That would be quite peaceful.
But I think you like more the heat than the cold. What do you like more?
The cold is quite fine. These bones don't care.
Okay, before we sacrifice death, is there a question from the public to death? No? I like that. That's a good question. What is your last meal?
I prefer souls.
Okay, we have a question here also. Rudy.
No, not for that.
Okay, so why don't you come in front, Death, here, and see how we can sacrifice you. So grab that. We are all going to punch him.
Halloween. Halloween.
So, yeah, we are here to kill death before death kills us. So, for immortality! ¡Viva la revolución! ¡Viva! Okay, now death, you can go. And let's go for the questions. We want people. This is a fantastic panel. We have incredible people working on different projects. I'm so excited to be here. This is our family. So, questions from our family members. Okay, all the way. Okay, first one that is closer.
Oh, this is more of just an observation. I thought the points you made, Joe, were just truly deep and profound and hit me in my heart. I think all of us struggle with various things, and including lack of influence and frustration and ego and all that. But if we can collaborate and basically put our private concerns aside and think bigger and think longer range, we'll all be better off. So thank you for that.
Yeah, a lot of people in this room, the people who put RAADfest together are from People Unlimited, and we have the strength and the focus to do that because we have one another. Okay, so we're not solo agents. It's tough to be a solo agent, immortalist in this world.
Okay, we have a question here in the back.
Thank you, Joe. It's very inspiring to hear you all the time. You are an understanding human being. Unbelievable. I treasure you. You talked recently about it's going to need a new faith. That touched me very deeply. Can you explain a little bit to the audience what you mean by having this new faith?
Yeah, I think a lot of us have felt like, something was going to happen. There's going to be a particular breakthrough before now, and have gotten discouraged. Horrible things happen. I lost Bernie. I mean, that's the fucking worst thing that can happen. And so there's a faith that's basically we're externalizing. And this is the faith we learn. We actually learn this from religion, which is really we're like, and I don't mean to. We do, but even if you're going to keep your religion, we have to modify. Okay, we have to develop here, and so there's a faith that's innate. It's innate to the human body. It's a core gut feeling of life. It's not really dependent on what's happening out here. It's not really dependent on, is this breakthrough going to happen? Is this piece of funding going to happen? It's that faith that's just core in our bodies it's not part of our mentality. We can't really measure it. It's not dependent on anything out here because good things happen and bad things happen. And that's how we keep going. Because it's something physical. It's something. I wish Rose was here, Michael Rose. He probably have something to say about it because it's something innate to the human form that we're tapping into. It's not doctrinaire. It's just organic.
Okay, we have balloons for the people with questions. It says "Stop aging" in Spanish. Stop envejecimiento. So you have a question, or you want the balloon? Or both.
What can we do? What can we do to double this attendance of RAADfest this year? Double it again next year. Double again. Keep doubling it every year.
Yeah, thank you. We need help. We need help. We work our asses off. We have about 700 registrations here. It's a little bit more than last year, which is nice. But that's really what we want to do. That really is our vision. And we need help. Like we need other organizations to see this vision and share this opportunity with us, and we need people to do it. We need people to bring people. And we say this every year, and the truth is it doesn't happen. So we need a shift in mindset, and I mean part of it is just me being more direct like I am here. I don't think we've been this direct. But we need help. You know, people come up to us and they go, we're going to help, we're going to do it. And a lot of times they're marketers and they're just selling us their services. That's not really what I'm talking about. I'm talking about people who want to share this vision and this mission with us. Yeah, because it's for everybody actually.
Okay. Here another question in the front.
Yes. So this room is full of change agents, right? When I worked to create the Health Span Action Coalition, which has 275 cross-sector organizations, I worked with a wonderful operator that helped manage Prop 71 and Prop 14 and got $15 billion of California taxpayer money for an agency for regenerative medicine. That was the work of advocates, and that was the work of narratives of people, right? Narratives of people. So before I took this on as a stem cell advocate and a background in law, a person that inspired me was a young woman, Sabrina Cohen. She became a quadriplegic in an accident when 14 years old from Miami Beach. I met her at the Miami Project to Cure Paralysis when I was giving a talk. She looked just like Sandra Bullock in a wheelchair. She became the first associate director of my organization, the nonprofit organization that created the World Stem Cell Summit. And before we launched the Health Span Action Coalition, which was founded on an idea between Aubrey and I to bring this world together, health span should be a fundamental human right. I asked Sabrina, who has gone on and a top realtor and created Sabrina Cohen Foundation, Adaptive Beach Days in Miami Beach. Sabrina, does immortality, does a long life, a long lifespan matter to people in your community, meaning the SCI, spinal cord injury community? Yes, Bernie. We in our community know we're aging faster. We know we are going to die young for all the things that we go through day by day. And I will join you, and the spinal cord injury world will do it, be a part of this. This is cross-sector, and and you are the spark. I mean, I am really inspired by being here. I was nervous about coming to this because I heard about RAADfest and it might be like a rave and, but this is really the real deal. So I just want to say how inspired I am and proud to be associated with you.
And Bernie, those words coming from you are very important to all of us, and all you have been doing for so long. So, okay.
Yes. First of all, Bernie, I'd like to thank you for the heroic work that you and Melissa King have done over the decades in advocating for stem cell research, which is a crucial pathway toward life extension and curing deadly diseases that could kill people in the meantime. And I also wanted to echo what you said in terms of encouraging people to find initiatives that already exist that they could contribute to. And this also goes along the lines of what Joe articulated, because even when I was looking out to see how I could contribute to the field, I was thinking about what endeavors were already in progress that needed help. And you are important, as Joe said. One person can make a difference within that collaborative approach. Like with LEV the game, I saw that there was a previous team that had tried to develop it but didn't have the funds, and I didn't want that project to just languish. I didn't want it to disappear down the memory hole, since there wasn't even the source code for the original game remaining. So I brought it to a playable state, but I needed another individual, Juraj Stern-Vukotic, to program it. I needed the community to provide feedback. Likewise with the Transhumanist Party, when Zoltan Istvan founded it, he ran for president, he had the Immortality Bus campaign. In 2016, he decided he wanted to move on to other endeavors, so he stepped down from the leadership role of the Transhumanist Party. Really, he's more like a serial startup founder than someone who keeps an initiative going for years, but he wanted it to go on for years. He wanted it to outlast his tenure, so he asked me to be the chairman, and I decided, well, this is my opportunity to contribute to something longer lasting and perhaps something that would last beyond my own tenure as chairman. So think about those kinds of initiatives, those that are already in progress, that you could make a difference in and you could carry to the next level.
Okay, good point. Ilana, a balloon and a question.
I want to ask a question for the balloon, so thank you so much. No, but so you were talking, Joe, about numbers matter, and which is true. But there's something about passion and having such a conviction of moving this initiative forward. And I was wondering if this is a good time maybe to tell all these activists here what it takes to create RAADfest and how even one more person that jumps in the way that they can contribute can make a difference. Because I don't know if people here know what kind of team you guys have and how long it takes to actually produce this event. So maybe you can give a little bit of detail.
Yeah, we have a really small, really good team of, I want to say, Michelle Andrews, Gene Weidman, Mariana the cellist, and Daniela Escobar is the team with Gennady. And, they're working on that, most of the year. And so, but the vision is of a platform, and that's where we can do something that doesn't really have a limit in size. And we basically do it on like a zero budget. Like we basically do it on there's no extra money in this. So we, and Jim doesn't get paid, and I get paid a little bit, that's why I'm doing messaging strategy for, boring companies, but which I try to help them. But yeah, so and this is a little bit where we are, because we're on the cutting edge of longevity, right? I mean, and this is where the space is right now, and it doesn't need to be. Like we could have volunteers that have superb capability that could really, as you're saying, really expand what we're doing. We could have a chunk of money that isn't even that big of a chunk of money that could change how we function. So, and it's time for that. Honestly, it's time for that. So you guys love RAADfest, we love RAADfest, but we don't want to keep just doing it like this. We don't. We want it. We have to go in a more. I want to say bigger, but it's not just about bigger. It's just we have to expand. That's what has to happen.
To add to that, one person with a passion is more powerful than 100 people with an interest. So whatever we can do to increase passion in ourselves, we need to do that, and we need to bring more people in who have passion more so than interest.
Just let me add, we need to collaborate internationally. Like, for example, I'm very happy that every year I bring friends. In fact, here is a Spanish friend, Elena, and there is a Mexican friend, Doris, and then also Cody. Cody always brings friends. We have Mark Hamilton, we have Wallace behind. All of us bring people. We need to bring more people. We need to collaborate, and we need to keep this growing. And also in different places, because I organize something in Spain as well. And my public is mostly Spanish. And then some of those come here as well. Yeah. So we need to keep on collaborating and also experiment different things. Like we are doing this march in the centers of power of Spain. That is why I think it's so important that we have a rally for longevity in Washington, D.C. Washington, D.C. has to see this. As Gennady mentioned, the immortality bonds, yes. The coffin, it was a coffin in front of the U.S. Capitol. That actually should have had more media coverage. And, you know, the Coalition for Radical Left Extension can do it, and better, because I like Sultan Isvan, but it was a one-man show. Yeah. Here we are hundreds of people from all over the world. And then before giving you the word, Jim, because there are other initiatives we need to adapt. And I want to ask, because Cody hasn't spoken too much, where we are going to be next month and what we are going to do next month.
Yeah, let me just jump in just quickly. Look, we're not just like immortality entrepreneurs. We're not just like starting something and moving on to the next thing. That's not what we're doing. We're in this, and we're not going to quit on this, and we're actually not going to quit on you. And that's the difference. You know, it's one thing to have a campaign and do something and then disappear into something else. That's not what we're about. We're on this, and we've shown that, because 10 years is 10 years. We're on this to stay. That's why, honestly, that's why you should support us.
Well, we are here to kill death. Look at that, death there. We are here to kill you. And there are different approaches. So what are we going to do? Who are we going to meet? Where? Cody.
Okay, well, first I want to say Immortalis is kind of doing slightly what RAADfest is doing, but on a different stage. So RAADfest gives the scientists and the researchers of longevity the stage and the voice to the public. And Immortalis is giving the stage to those same people to move forward faster in what they're doing. Right. So next month, we are going with Jose he's going to introduce us to one of his friends in Argentina, Javier Milei.
The president of Argentina is going to meet with us, with Mark Wallace, Mark Hamilton Wallace, and Cody and myself, because we are proposing to have Immortalis in Buenos Aires, south of Buenos Aires. And we want competition also. The USA has to get in there. Of course, the USA has to be there.
Yeah, Argentina is one of 3 or 4 governments we're in talks with right now.
So, okay, Jim, here is the father of the beast.
Thank you all. Thank you. Well, great job, you guys. Great job. Thank you. Thank you, all of you. You're great. You're fantastic. I just want to make one thing simple here that I'm feeling about all this. And, you guys look great, by the way. Is that I want to keep it simple in the context that when we talk about companies and everything, helping and expanding, helping to expand, we're not talking about anybody stopping what you're doing. What you're doing is great. Cody, all you guys, Gennady, but make RAADfest your home in the context that make it your base. Make it that you're telling your people, all your members, right when RAADfest ends, that they need to be at RAADfest. Not just sitting back and waiting for your different meetings you have. Get them here. You're already doing that in some state, but this is really what we're talking about. This is the simplicity of it. This is how we're going to double. This is how we're going to triple. And in fact, everybody here, talking to your friends, your family, everybody, get into this conversation. Get into this life. Speak it up. And making it that simple. We're come many. Thank you, Joe, for your presentation. All you guys, like I said, you're amazing. But let's keep it also to this point simple that we talk to our people right when RAADfest ends. You're setting them up, and you do a great job setting them up now to be here. And it will be this. Then RAADfest will build something that'll be a springboard for all of us. It will build everybody. That's the point. By us focusing together and this initiative, we will build then everybody's initiative along with that. But if we don't have that one focus, we're scattered, and that's what happens. Thank you.
1, 2, and 3.
Yes, my wife is the president or vice president of ALICE, which is the Latin American Sociology Association. They have their conferences every 2 years. So what they do in the meantime is they have pre-ALICE conferences all over Latin America. There might be 3 or 4 or 5 of them in Mexico alone. And optimally, you want those people coming to your main conference, but in truth, they're not all going to go there. So what would happen is you would invite just a few speakers from the main conference to go to all these different locations. You're promoting it. My wife's the president of 10 universities in Mexico. We would hold one at our university. That would give us, we make it very inexpensive for the students to come. So the students, pay the expenses of the conference. And most of those, 90% of those students and other people don't go to the main conference, but you're still spreading the word.
Right.
Okay, so I don't even know where to start. To your point, what I tell my friends, just to simplify, is I'm going to the first largest conference in the world on longevity. Your presentation was right on. I'm a global marketing communication expert, and I'm not selling anything. I want to collaborate and help. I told Liz Parrish that I moved to Mexico because I believe that access to the things that people need are much easier being in Mexico. I speak Spanish, so I'm fortunate. So whatever is needed, I'm there. I'm in Tijuana. I'm right on the border. One of the things that I think is critical, in my opinion, of teaching at MBA programs, working in marketing all my life, is you need to think from the outside in versus inside out. What does that mean? When I talk to people, regular people, to your point, how do you double attendance? You need to get people excited and passionate. The people that I talk to about this concept are afraid. They're afraid. And what are they afraid of? You'll never guess. They're afraid of loneliness. They're like, if I outlive, I look at my mother, 2 of her friends just died. It's very sad. So when you're talking, when you're excited, because you've already bought into the concept, you cannot get through people. And I'll tell you this as a marketer for many years. People will just be negative, or they'll just shut down. You will not convince them in that way. So what's needed is, to your point, this is a time for strategy. This is a time to sit down and figure out all of the strategic elements of driving this movement forward. One with collaboration, one with messaging, one with outreach. Very clear, simple messages that can help people get over the fear. Look at the benefits, sell them on the benefits, so that you can bypass either misinformation or just get them thinking in a more positive way. And I'm also open to helping in any way with that. That is one of my expertises as well. But I believe there's so much that can be done at this moment. There's a lot of momentum, but it is a time to collaborate and be strategic so that you limit the resources, and with less energy, you can do more. And it's hard, but it's doable. You have amazing people. There's amazing people here that I'm just like, I've never expected to see all of this. I've been in touch with Jose. I knew about all of these conferences, but there's so much more that I think can be done just to move all of these forward. So again, thank you for that presentation. I think it was wonderful.
Yeah, thank you. Thank you for your comments, and we'd like to connect more with you. I do want to say that the more obvious opportunity is people who are already into longevity. Okay, we're talking about travel. We're talking about paying a registration fee. We're talking about spending a couple days somewhere. That's the obvious target. It is the transhumanists. Okay, there's actually a significant number of transhumanists. There's actually a significant number of longevity people. I'm not saying we shouldn't reach out, expand the market. We 100% want to do that. But let's mobilize the people who are already into this to actually, fucking do something.
I have some feedback that might help us improve our message. There's an organization called Vistage. It's an international organization of 45,000 CEOs in 37 countries. I'm a speaker to them on sales and also on longevity. When I start my longevity presentation, I'll ask, raise your hand if you would like to live forever. About half raise their hand. Then I'll say, okay, raise your hand if you'd like to live forever with great health, a sharp mind, unhindered mobility. Now we're at 3/4. And then if I ask, raise your hand if you and your family and your friends could live forever with great health, almost everybody raises their hands. So we might think about that, and somehow it might help us improve the message. The second feedback I get that's negative is, this is against God's will. God has determined how long we're going to live, and there's nothing we can do about it. So there's no need for me to get involved. I don't have an answer for that myself, but I will tell you my wife's Catholic, and that's how she feels. So there's two philosophies in life, determinism, that I just mentioned, that she believes in, and the other is free will. That means I'm in charge of how long I'm going to live. And number one, by what I do, but number two, if I'm doing good for the world, like I think I'm trying to do, then the man upstairs will see that and keep me alive longer. So I just wanted to mention those two main pushbacks, so it might help us improve our message.
Beautiful.
Yes. Just to piggyback on that, I know I did a little bit, but I do think we need to stop this like boxing match with religion. Like let's you know, if you believe in a God, just believe in a God who wants you to live. You know?
Yes.
Yeah, let's welcome everybody.
Yes.
It's actually for everybody. Because we want to live. It's not about an agreement in doctrine. It's not really about that. It's about human beings living.
Right. Yes. So I agree with you, Joe. I think determinism, irrespective of religious or philosophical roots, is a kind of evolved coping mechanism that various traditions of thought have developed over the course of millennia when mortality really was unavoidable because the technology just wasn't there. So people figured out a way to make peace with death instead of trying to fight it. The problem with that mindset is right now we are on the cusp of being able to kill death, but it will be dependent on our efforts. So any sort of determinism or fatalism will undermine those efforts, and we ultimately need the religious communities of the world to align with us on this. As I've said yesterday, religions evolve too, and we can hopefully influence them to evolve in a positive direction without requiring people to give up something that's valuable to them. So I'm an atheist, but I don't want to deconvert anybody. If you are a Christian or a Jew or a Muslim or a Hindu or a Buddhist, I want you to be a Christian or Jew or Muslim or Hindu or Buddhist who believes in immortality or radical life extension in this world. Yes, and that's how we can get to it together. And then we could argue about theology or philosophy all we want once we have immortality, and then we could actually drill deeply into what we believe and why without running out of time to do it.
We could argue about it forever if we wanted. So I don't understand the argument of it's against God's will for longevity, because the Bible says we're created in God's image, and I haven't heard about God dying, so...
That's right. It's the Hebrew is B'Tselem Elohim, right? So it's in the image of God.
The gospel of longevity, Jim. Okay, we have a question here, Bob, and a balloon.
Yeah, I think we need to walk our talk. I had my 50-year service celebration at the Boeing Company about 3 months ago, and it happened to be in the middle of one of my fasting periods. I thought, am I going to break my fast for this? Hell no! You got to take charge of yourself and say, I'm going to be different. You are different, you know? Do the things and do not pay attention to what the masses who follow the sheep expectation follow. You're going to retire not to death, but to do more things. There you go. When you have your 50-year fast, if you're going to fast, you know, don't stop.
Okay, we have time for 2 more questions. And also remember, after this, we are going to have a very nice meal. Life is also to be enjoyed. That is why we want to be immortal, because we want to have fun. We have to enjoy life. So the food will be nice, but we have time for 2 questions. One question here. Taylor.
Yeah, I have a couple of questions. One is actually for you, Jose. In Madrid, are your speakers that you have planned, are they actually going to be there in person and not over Zoom? Because that's a far way to travel for a lot of us. And another is for Gennady. And we've had a lot of exciting protocols announced here on the main stage and in some of the clinics. And are you considering incorporating any of those into your game as things that might pop up if you're advocating for anti-aging?
Okay, the first question, yes. I like to do like in RAADfest. The speakers have to be there because I want also the people to talk with the speakers. This is important. And yesterday there was a beautiful VIP reception with the speakers and those participants who wanted to go. And so I want them to be in touch. There are certain exceptions. Last year I had Brian Johnson, but Brian Johnson tries to avoid international travel because of his circadian rhythm and his sleep. And this year, so he didn't come in person. He was by Zoom. This year, I also have one person by Zoom who is George Church. George Church is no longer traveling internationally. And so is Ray Kurzweil. He doesn't travel internationally. But otherwise, all the people are coming to Madrid. Yes, this is a real conference. And I have tours for them. I want them to enjoy life, to see how beautiful life is and how fantastic the Mediterranean diet is.
Yes. So, Taylor, to answer your question, I'm, of course, still open to feedback on LEV, the game. We're currently on build 0.91, so we're not at 1.0 yet. The game is open to iteration and refinement. Now, in terms of how specific do we want to get, there's a reason why I had a singular selection for fitness regimen and a singular selection for improved nutrition. It's supposed to be whatever you think works or whatever works for you or whatever ends up working in practice. And the reason why I did that was because I wanted to show at least the approximate scale of effect. So if you engage in the best possible fitness regimen, whatever that happens to be, that could reverse your aging somewhat, that could rejuvenate you somewhat, make you healthier, make you live longer. It's not going to make you live to 150. Likewise, if you have the best diet in the world, it could help you live longer. It could help you avoid certain diseases. It's not going to make you live to 150 or 1,000 or 1,000,000 years. So within the game, I'm less focused on being minutely accurate. I'm more focused on accuracy in relative terms. Like, what is it that you would really need to do? What kinds of interventions to reach that indefinite, open-ended lifespan, immortality, if you will. And that's why I not only include the rejuvenation therapies, I include other causes of death too, and the opportunity to fight those, which you will have more time to do if you're ageless. So think about all the risks facing humanity and the effect size of each of them.
If there is one question, and also I want Death to ask a question. Death, come here. I want you to ask a question to our panelists. So think about that question while we have a quick one, right? Quick one, and then Death, think about it, because it is a matter of life or death.
You said we don't want a boxing match with religion, but what if religion wants to fight us? And particularly, if they oppose cryonics, for example.
What if those who are religious or of certain religious persuasions want to fight us? That's the question. Joe, go ahead, and I have some thoughts too.
Yeah, it's still not a good strategy. Okay, there's a lot of people who it's just not the way we want to go. There's a lot of people like in the middle ground that we just want to bring around to our to understand what we're doing, and that's what we need to keep going with. And
Yes.
It's still the best way to go.
And my view is fight the dogmatism. Don't fight the core of their faith or their religious tradition, but fight against the fact that they want to impose their views on others. And I have a rather strong libertarian streak, small l, not capital L, but I believe in individual freedom. I believe in your right to make the choices that you want to make for your life and your well-being as a patient, as a health and wellness enthusiast, as a cryonicist. So the argument from individual rights will resonate with a lot of both secular and religious individuals, and you can appeal to a lot of people who might even be, say, fundamentalists, but tell them, oh, well, what about the U.S. Constitution? What about the Bill of Rights? What about what the founders wanted individuals to enjoy as their freedom?
Oh, okay, now,
Hold on,
death.
I want to say something too.
Okay,
on that.
quickly.
Also, you got to look at the silver lining of people fighting us, because that's actually progress. Because first things are ridiculed, then they're violently opposed, and then it's widely accepted as truth. So we're making progress when they're fighting us.
Yeah, very good. That's Sir Arthur C. Clarke who said that. Anyway, death has the final question, so it must be an immortal question.
I realize I strike a lot of your hearts with anxiety and fear. How best do you want to ameliorate that?
I didn't understand.
Well, I don't know if I want to alleviate the fear of death as long as you exist, because fear can be a powerful motivator. Now, I've been asked this question a lot since I published Death is Wrong. Well, aren't you doing all this just because you're afraid to die? I don't deny that I'm afraid to die, but I have to go deeper than that. And I have to ask, well, if I'm afraid of something, why is it that I'm afraid? What is it that I'm afraid to do or become? I'm afraid to lose something good. I'm afraid to lose life because life is good. Why am I afraid to lose life? Because I love life. So I think fear of death isn't something to be ashamed of. Fear of death stems from love of life, and love of life is something that should be embraced.
Well, sometimes people argue with us that we need death to some kind of catalyst in life, and we really don't. And so I would say, let's not make death the catalyst of our movement. I think that what Jim was talking about on the main stage, I think it was opening night, about being drawn to consider ourselves pre-human, about being drawn to what will be our true human state, which is what would be considered heaven on earth, but it's really what earth should be.
Yeah. Don't take it wrong, but.
Gennady, at the same time, we need to focus on joy, abundance, and what we're creating.
Beautiful, Jim. Actually, when people say that what gives meaning to life is death, that is so stupid. It's like saying what gives meaning to marriage is divorce. No. What gives meaning to marriage is marriage. What gives meaning to life is life. More life. So, we are going to launch. We go and take some pictures because this is for immortality, and he's a very good-looking death. So have some pictures with death also. Happy lunch.